my name is Morghan Lucas and I came from Australia to do a PhD in Barcelona in the Center for Genomic Regulation with Dr. Eva Novoa in the Epitranscriptomics and Dynamics laboratory. I have a background in biomedical science and now I'm studying the third year of my PhD in molecular biology. My research project is understanding how metabolic disorders such as diabetes can be transmitted from fathers to their offspring through the genetic material found in sperm. I'm particularly interested in how diet can alter the sperm and then transmit this information about metabolic disorders to the developing embryo. If we can understand what is the mechanism and when it's important during sperm development, we can prevent these genetic disorders from being inherited. When I started my PhD at the CRG, they gave us a training course on how sex and gender differences can affect our research projects, and I knew absolutely nothing about this area before. It got me more and more interested in how gender imbalance can affect our everyday lives without us even noticing it. And I became so passionate about the topic that I joined the Gender Balance Committee at the CRG, and I started to consider myself quite educated on the topic. But it wasn't for another year that it dawned on me, my whole research project and field is grossly imbalanced. There is a very strong sex dimension to my own project that went unnoticed for 2 years. In the field of metabolism, we often work with mice, and it's known that female mice are much more complicated to work with because of their hormonal cycles. Not only have I been told myself by many researchers to work with male mice, but my field publishes exclusively on male mice. And yet we call it paternal inheritance, when we should really be calling it paternal inheritance in male offspring. But what about the females? How can we draw conclusions about metabolic disorders and paternal inheritance if we don't also study the female cohort? If we aim to study and treat metabolic disorders in humans, we should do that for the society at whole and not just for the male population. So when I finally had this epiphany, the project had already started, but I still spoke to my PI, Eva, about my concerns. Although it was no longer feasible to include females for every condition that we set out to test, she was happy to let me include a female cohort where possible, even if it meant more time, money, and animals. If we had considered this earlier, maybe it would have been possible to change our experimental design so that we could include both males and females without necessarily having to double the amount of animals and work that we had to do. I believe that a better gender balance in academia, referring to having more women leading research teams, would diversify the perspective on research topics and lead to the inclusion of sex as a biological variance. Perhaps if I had more female researchers as role models, or even researchers around me that question how the sex and gender dimension affects our research, I myself might have noticed sooner just how sex-biased my own research was. It can be difficult to question the norms of research, especially in published works which we often believe to be infallible, but the gender and sex dimension of research is not always obvious. That's why we should be always asking ourselves and others if our experiments and results can be influenced by gender or sex.